Hello all, I am Zane Walters, here on behalf of the Molecule Monitoring Lab at Grand Valley State University and the Kent County Health Department, discussing our method of sequencing SARS-CoV-2 in wastewater using amplicon sequencing. Now, this project has two goals. Firstly, to develop a standard procedure for this method of amplicon sequencing, and second, to establish a method that is both cost-efficient, reliable, and produces results in a timely manner. This process relies on our partners at the University of Missouri, namely Mark Johnson, who we work with directly, who does our sequencing for us using next-gen Illumina sequencing. Now, this sequencing is limited, firstly, is most accurate with amplicons of 600 base pairs or less, and requires us to use primers, which include an adapter sequence, allowing our amplicons to bind to the Illumina plate. Our sequencing focuses on the receptor binding domain, or RBD, and on more recently, the nucleocapsid protein. Between these two sequencing portions, we are able to identify most, if not all, COVID-19 variants. Our process is four-step fold. When we have samples with a gene copy of more than 9,000, we move on to sample amplification to make sure that we have enough sample to be sequenced. We do this with a nested PCR process, which will be detailed on the next slide. Once these samples have been amplified and their adapter regions added, they are sent off to the Johnson lab for their Illumina sequencing, which then returns to us raw sequence data, which we process through our Python program, which compares these sequences to our variant dictionary, producing us a list of both the present variants in every sample, as well as the proportion of those variants in each sample. We have two amplicons for our RBD sequencing. The first converts the RNA into DNA. It includes a reverse transcription step. And the second uses a smaller amplicon, using the primary amplicon as its template. And this smaller amplicon is 547 base pairs in length and includes the adapters, allowing it to be very accurately sequenced via Illumina sequencing. And here we see that same amplicon confirmed by gel electrophoresis, which we do before sending it off to the lab just to make sure it is amplified correctly. Here we see an example of a normal type of printout identifying both the variants present in every sample as well as the proportion in which they are present. We then use that information and report it out to our partners. But here you can see an aggregate graph detailing for every week since we have started sequencing both the variants present as well as in what proportion out of 100. And this graph is wonderful to see both the fall off, the decrease in the BA lineages, as well as the rise of XBB.1.5. And we are rather excited to report that in our most recent sequencing from the start of July, we have found both XBB.1.9 and XBB.1.16 in our samples, confirming that this process is able to also identify those. In short, this processing sequence compared to ddPCR from GT molecular kits, as well as whole genome sequencing, tends to be more cost-effective per sample, gives us our results in a more timely manner, as well as does not require re-extraction of RNA, and allows for relatively easy processing as well as accurate results. Thank you all for your time. I will be at my poster from 9 a.m. to 11 a.m., and I'm happy to field any questions you have then. Thank you.